All right, everybody. Thank you for joining. We're going to get started. My name is Mehdi A. I run the DNA sequencing facility at the Cincinnati Children's Hospital.
Has the conference.
I'm going to be talking about the genetic technologies that we have in the PCBC cell characterization core, and in particular, which technologies you can choose, how you can design your experiments, and how you can prepare your samples to get the best data possible. My goals for today are to present the technologies available in the core facility, for you which technologies can be applied to generate specific types of scientific data. I will go over four. Briefly show
has left the conference.
You see the data returned by each of the technologies.
Just a quick note for those on the call that at the annual meeting next week, there will be a follow-up session where it will be more the bioinformatic analysis of the data for which Mehdi is going to be introducing assays for today. Just to let you know, there is a follow-up next week.
So there are four technologies that we use at the PCBC cell characterization core. The mRNA-seq, the miR-seq, the CNV-SNP, and the methylation 450 assays. miR-seq and mRNA-seq
join the conference.
Oh, hi. Who's joined us?
The Illumina HiSeq 2000 and the SNP, CNV, and methylation 450 assays are run on the Infinium HD microarrays and ScanV. Quickly to make sure that everybody is on page with how next generation sequencing works. It's not just a matter of throwing DNA on a sequencer. You need to prepare that DNA for the ability to be sequenced by creating adapters into what is called a sequencing ready library. Then that library goes onto the sequencer and sequence data can be obtained. I'm going to go and turn over the technologies that we use. I'm going to start with how to generate transcriptome profiling using mRNA-seq. mRNA is meant to generate gene expression profiles by sequencing all the transcripts in a quantitative fashion. Quantitative fashion means that the more reads that belong to a transcript, the more expressed it was in the cell sample. In the example here, gene A and gene B each have a different number of reads that align in front of their exon, and you can see sort of qualitatively that gene A is a lot more expressed than gene B in this experiment. This is what I mean by quantitative fashion. The advantages of mRNA-seq, the major ones, there are two of them, is the potentially unlimited dynamic range compared to microarrays. If considering here a gene that have a very low expression level, it's very difficult to distinguish reads that align to that gene's exons from the background. There isn't a lot more. So the expression level for that gene could come up as very low in a first analysis. But you could sequence the whole transcriptome at a deeper level, and then all of a sudden, the reads will start to stack up over these exons in a more significant way than on the background. Even though the background would get random reads scattered around, the exons would have a much higher number, etc., etc. So this way you could sequence more and more and detect very low expression genes. The second advantage of mRNA-seq is the ability to quantitate alternatively spliced transcripts. When align reads to mRNA, there are some key features in at the gap
has a conference
spliced out, and by analyzing the reads that fall across the exon junction, you could actually identify whether an exon was skipped or an exon was added, thereby allowing you to detect more than just the expression level, but at the differentially alternatively spliced transcript expression level.
Having the conference.
So how mRNA-seq work? one of the issues is that total RNA from cells contain up to 99% of ribosomal RNA. Ribosomal RNA are the functional parts of the protein translation complex. We're not interested in quantifying ribosomal RNA for transcriptor profiling, so we would like to get rid of it before we sequence the messenger RNAs only. One of the features of messenger RNA that we use for this is the fact that most of them are polyphenylated at their 3' end. So as we take total RNA, process it over oligo-DT magnetic beads, and fish them out that way. I think I'm having problems with my cursor. Never mind. I can't point anymore. So after isolating the full-length mRNAs, we're going to fragment them, and before reverse transcription and actually ligating the adapters, as you've seen in the NGS process earlier, and sequence them on their HiSeq 2000. After that, the reads will align to the entire genome and fall in exonic locations where another software will be used to quantitate them. So how do you best prepare RNA for mRNA-seq data? We're going to do that for you, and if you are sending your samples through the PCBC C4, it's better to load cells in Trizol immediately upon harvesting and then send them to us. We'll use the aqueous phase, which contains the RNA, to extract it, and before running the experiment. It's also to have a good extraction for mRNA-seq because we're going to use this poly-A selection step. So it has one advantage and one disadvantage. The advantage is that it's not sensitive to DNA contamination. If we isolate a little bit of DNA coming from the interface of the Trizol with the RNA, will not recover this DNA after the poly-A selection. However, DNA that is present in the original RNA sample could interfere with spectrophotometric quantitation. That's why we also use spectrofluorometric within a specific dye to quantify the samples that are extracted in our laboratory. The disadvantage is that RNA integrity is key. If we want the 5' end to stay attached to the poly-A in order to be sequenced because the transcript is pulled by its poly-A tail. So we really need to have no breaks in the RNA in order to not bias the data towards only sequencing 3' ends, which are collected with the poly-A. So how do we design a transcriptome profiling experiment? There are 3 parameters that should be taken into account: the number of reads that we need, the level of information that we want to obtain, and the cost. I am taking the example of a simple 3-exon gene that has low expression
has joined the conference
and an equal amount of 2 of the isoforms expressed. With 10 million reads generated for a cost of $350 through the PCBC core, there's no mouse. You will see that there's a slight you could have a slight difficulty with the low expressor in terms of quantifying the 3 different exons individually, which are present in this sample. But if you generate 30 million reads for $550,
conference,
the number of reads that accumulate on the exons that are common to the 2 transcripts is all of a sudden, coming up to be clearly double the amount of exon that is present in only one of the isoform, and which would allow the software to actually quantify each transcript separately and accurately. The accuracy would be missed in a 10 million read experiment. So it depends on the level of information that you need to have and the money that is available for that experiment in order to order the right number of reads. Now, in terms of replication, the rule of thumb for stem cell research that I would communicate is that technical replicates are not necessary. but biological replicates are absolutely needed. If there's anything, if hesitant about what to do with the biological replicates, I would recommend that you follow this rule. It will allow your statisticians to have a wider range of possibilities for comparing samples. The rationale is that what you really know is if the difference between two samples that we're interested in comparing is stronger than what we expect to see between two samples that are replicates. So if you are able to answer this rationale question, then you can decide whether you need biological replicates, but we are here comparing usually, progenitor cells, iPSCs, and they tend to have similarities, not like brain or liver, and we recommend using biological replicates for that. Now, how do we QC mRNA-seq data? There's a very nice software developed here by Philip Dexheimer that I would like to show you how and illustrate how we use it. So on the left and on the right, I have two samples. Each have a similar number of reads. 11.7 million here and 13.7 million here. These are both mRNA-seq experiments conducted in single read 50 fashion, and you see at the level of the alignments that the number of strands that align to exons, spliced exons, introns, spliced introns, and intergenic regions is very much the same: 85, 6.5, and 8.5 in one case, 88, 5.4, and 6.4 in the other. So really,
has joined the conference.
You really wouldn't tell, you get both results from both samples, and if you don't do type of QC, it looks all fine, and you would basically not do that. You can't compare these two samples. However, Phil has developed an analytical tool that tries to correlate the number of 5' bases in the transcriptome profile with the number of 3' bases covered within a single gene. It's displayed here in the red line, as the blue lines represent all the other samples in the data set. If you see on the right side, that sample has actually a strong deviation from the rest of the analyzed data in terms of how much of the 5' ends are connected to this 3' end. And we have developed this
has left the conference
ratio that we have empirically determined to be above 0.7 in order to have some data that you can be confident about utilizing. If you have degraded sample and there's no other choice, you could compare samples that have a low 5' or 3' ratio, but I would recommend that they be all aligning along the same lines in terms of how much of the 5' connected to the 3', to avoid biases. So that completes the presentation of the mRNA-seq profiling. I'm going to move on to the microRNA profiling using mere sequencing. MicroRNAs are transcribed from the microRNA genes by
left the conference
and processed inside the nucleus into pre-mRNAs, exported to cytoplasm, where enzyme that we heard a lot about, dicer, prepares them their mature form, to which the RNA can be wound into two forms which have different effects in the cellular mechanism. So because the mRNA targeting and the degradation of RNA roles that these microRNAs have, it is sometimes important for people to quantify the amounts in their cellular process. How do we do that? In the core, we take advantage of the fact that the DICE microRNAs, the mature forms, have a 5' phosphate and a 3' hydroxyl, whereas other RNAs are usually capped and don't have these phosphorylation statuses at their ends. We use a specific ligase, the T4 RNA ligase 2 truncated, which ligates only pre-adenylated oligos to the 3' end of RNA. This allows us to, in the total RNA, to make mir react with a 3' oligo that has been adenylated and ligate to the 3' end of the mirs. After that, we use
joined the conference.
A ligate 1, ligate oligos to the 5' end of RNA, and what you see is that we have achieved at the RNA level library construction that we would with DNA that I have described early on for the process of next generation sequencing. So using RT-PCR with primers that match the adapters that we have just ligated to this RNA, we can generate double-stranded DNA library from mostly microRNA, but it will also ligate some small RNA forms. So we do a size selection step. We let microRNAs are anywhere between, 18 and 27 bases at most before we throw them on the sequencer and map them to MEAs precursors. So it's automated. This size selection is done automatically on a Pippin prep instrument. It's pre-gels and computer controlled, so we can actually separate our input represented here in red in the bioanalyzer trace. We can separate the red input from dimers and other small RNAs. The mirs that we want, which we know are right here at this specific size. It's often difficult to separate, other small RNA forms, probably peewees, completely from the red input. But as you see in the blue input, it's pretty much clean of any other forms of RNAs that may have reacted in this mir sequencing process. So how do you prepare RNA for mir sequencing? The same way that you do prepare your cells for sending to the core, lysine triazole immediately upon harvesting is what we recommend. And we will take care of isolating the RNA in a way that preserves the very small fraction of the RNAs for compatibility with your sequencing. Because of various specific ligation steps, we can make two conclusions that, again, DNA is not going to be sensitive to DNA contamination because DNA is not reactive to this ligation step, which is necessary for library preparation and for sequencing. Again, it interferes, however, with quantitation of your RNA. That's why we use spectrophotometric methods. But RNA this time, RNA integrity is not as critical as for mRNA-seq, although we would much rather run with clean RNA than dirty RNA. So we still suggest a bioanalyzer RNA integrity number strictly superior to 8 before processing the sample. But if you're a member of the PCBC, you don't have to worry about that. We will make sure that we have a good extraction for you. So how do you design a microRNA profiling experiment? In terms of number of reads, 2 million reads is usually plenty for microRNA profiling with good RNA. We've noticed that if the RNA degrades and we see more small RNAs that are reactive to those specific ligation steps, we may want to increase the number of reads in order to maintain around 1 million reads that are actually going to go into the microRNA profile. Now, one exception would be if you're searching or suspecting the presence of novel microRNAs in your sample. Up to 10 million reads can be useful if they are in low expression level, which would increase chances of actually finding them above the background of anything that does not align to the precursor microRNAs. In replication, the RNA profile and microRNA profiles are fairly similar in terms of their distribution of low levels and high levels. So we recommend the same a replication as for mRNA-seq. It is not necessary to do technical replicates, but we recommend up to 3 biological replicates, so you can use a t-test for your estimation of expression levels at the statistic comparison later on. The quality control, the quality control that we use for microRNA is based on the read size after adapter trimming, which will then reveal, because we are sequencing the full-size microRNA, which will then reveal how frequent the proportion of reads that are around the size of a microRNA, which is most abundantly at around 22 bases here. And we compare the number of raw reads that were generated in the experiment. This is just a sequencer calibration issue. To the reads that have a size above 15 base pairs, which would be a function of how good the RNA is. And then out of these reads represented here in the histogram that are above size 15, how many of them actually align to base precursors, and we expect, high numbers here depending on how good the size of the library was. To do contrasting example, generating even more reads in this sample has much fewer reads that are below 15, so that means we had a lot of reads that were very small, and they're basically too short to be aligned to anything. Only a third of the data, probably just about 1/4, was actually recognizable, and only a fifth of that fourth is aligning to the mirror base. These would be these species right here. So then end up with a microRNA profile that only has 500,000 reads, which is on the low side. These species on the left and right are not microRNAs. So I am now switching to the genome-wide methylation profile using the Infinium methylation 450 array. We use the methylation 450 array from Illumina. The probes that are assayed on the array are mostly CpG islands, but they are out of the 450,000 loci across the genome. There are some non-CpG methylation sites that have been identified in stem cells that are useful to be assayed. Globally, the array captures more than 80% or about 80% of the genes in the human genome, with probes that are representatively illustrated here in terms of the distribution in the 5' UTR first exon gene body, 3' UTR, and then of course the promoter is where we are usually interested for the methylation detection, as well as non-genic CpG islands. 90% of the ones annotated in the genome are captured at least with one probe in this array, and the average distribution looks like
has left the conference.
Shores of the island, but most densely around the CpG island. So how does this methylation 450 assay work? You basically differentiate between the methylated C and the unmethylated C by bisulfite conversion, which will transform your genomic DNA into the methylated C is protected, whereas the unmethylated C is transformed to uracil. And after a round of whole genome amplification, you then copy these natively to C if it was originally methylated, and then to T if it was unmethylated. Apply the assay by hybridizing this genomic DNA that has been bisulfite treated onto an array that contains two types of probe. One probe for the unmethylated form will have a C for the G of the CpG island, and an A for the unmethylated form. and then you perform a single base extension past that locus, and any extension of this species will get you a signal, and if your locus methylated, it will not hybridize here at this level to the unmethylated probe, but it will instead hybridize to the methylated form of the probe, which has the full CG, where now the single base extension can occur and color at that location can be detected on the array, and then you put the array through the high-scan scanner in order to get a representation of the entire surface to hold 450,000 probes. So to give you an example of what methylation 450 data can look like, when you zoom into specific areas, I've chosen two iPSCs that are biological replicates. I'll call them A and B, an H9 ESC cell line, and as well as a somatic cell. And I've looked at two genes, one of them nanog, and we see that whereas the methylation pattern for all the progenitor type cells looks identical, it's quite different in a somatic cell around the promoter area of nanog. On the other hand, for a gene like PDGFRA, there is no real noticeable difference. I mean, this is not a very, this is just an illustration for a very, broad example, an effect that I can detect just visually, but I don't pretend to say that quantitatively this would make sense. I do not know that this is something that is obvious, but I understand these genes are important in PBSC for the consortium. So how to design a methylation profiling experiment? I'll illustrate technical and biological replicates are usually unnecessary. If you look at the level of reproducibility of biological replicates, it's really tight when you try to compare methylation profiles between different lines of an iPSC, where there's going to be a lot of difference. So because here the effects are quite large, biological replicates are not really recommended compared to mRNA-seq and microRNA-seq, where the differences between the lines are going to be a lot tighter. I am moving on to the last part of the fourth assay that we perform in the core, the molecular karyotyping using SNP arrays. In SNP genotyping, I'm sure everybody's familiar with the detection of a B allele signal and an A allele signal, and 3 clusters that will get you a SNP genotype described as AA, AB, or BB in a normal diploid state. When you're doing SNP genotyping with a copy number variation, here 1 copy, you have now you notice that you start getting clusters that have only 1 allele, so the intensity of representation of the B allele are basically cut in half, and similarly for the A allele. When you have 3 copies of the gene instead of 2, you now have 4 possible genotypes: a BBB that is 50% higher than BB, an ABB which falls anywhere in between AB and BB, an AAB which is the opposite of ABB, and a AAA, again at 50% more signal in the A allele than B. That if we try to model the position of these clusters in polar coordinates, we can take 2 views. This R, because all these in normalized B allele versus A allele signal, all these clusters which have 2 copies are on a circle, and here the same way, and here for 3 copies the same way. The R, the radius from the center, is actually proportional to your copy number, whereas the angle here is the proportion of the A allele to B allele. When it's 45 degrees, it's 50%. When it's 90 degrees, it's 100% B. So what we do is we transform these polar coordinates and plot the data in a genome-wide fashion of the normalized R and theta values. We normalize theta, which is usually between 0 and 90. We literally do it. We between 0 and 1, and then we normalize R versus an average R of control samples at the same SNP, and we take the log base 10. And then we draw these plots. So in the theta plot, or B allele frequency, you can recognize the SNPs for one individuals that are the AB genotype compared to the ones that are AA and the ones that are BB. And when you do the log of the radius compared to what a bunch of other samples of the same sex, usually, so that you can do it on the sex chromosomes as well, you see that a number of 0, which would be a ratio of 1 once it's logged, would indicate a no change in the copy number here, a fairly straight line around 0. And we start getting aberrant chromosomes. Here, this is an example of an aberrant chromosome 12. Both graphs are starting to change, meaning R and theta, according to the same rules that we've seen in the SNP graph. So these set of plots are, again, here is a set of SNPs along the chromosome. In this area now, you have SNPs that have the AB genotype. Here, SNPs that have the ABB genotype, which is concordant with a gain in the R signal, indicating the presence of a triplicate, 3 copies of that chromosomal location. And also, something that happens here is that you no longer see any heterozygote, and you see a loss in the R ratio, indicating that there's only 1 copy in this area. So the two graphs work in conjunction to help you distinguish, both at the genotype level and at the intensity of the probes, what the number of copies is present in the sample. It's a little bit more accurate than array CGH, where the allelic ratio cannot be estimated. What's happening here is this is the centromere. There are no probes on the array for the centromere, so you have a disconnection. You do not assay around that area. So another example, something that we can detect for the PCBC is really big aberrations, like the one we were able to observe on the karyotype, where a large chunk of the long arm of chromosome 13 was deleted. So all of a sudden, there is no longer any heads because all the possible genotypes are either A or B, and the log R ratio falls as expected compared to the rest of the chromosome. It's a little bit noisy, but it's very, very clear. So how do you design a molecular karyotyping experiment? There is no need for technical or biological replicates, again, because you estimate delete. These are very clear signals. And in terms of normalization, the control for sample-to-sample variance are already in the software. This is what you use to calculate the log R ratio. What we recommend is that you save the pellet from your starting materials when making the iPSCs, and then you should run your starting material and a few cell passages. So I don't know, Carolyn, 30 passages is what?
We work between 15 and 25 is our goal, so something in that range.
So you could, because the idea is that each passage increases the chance of chromosomal abnormalities. So you want to know if the chromosomal abnormalities detected in your iPSCs was not present in your starting materials. That's why we recommend saving it. And we are building a database for the small CNVs, which
has left the conference.
they're too small in every sample. So this is the lack of reproducibility aspect of this method, is that when the CNV is very small and we see it only 2 or 3 SNPs, it's very, very difficult to confirm it. So what we do is we put these into a database that we keep separate from the CNVs that we know are clinically relevant, in hope that later to be able to analyze this and maybe see some CNVs that are more common in after passages of the atlases. So for both
has left the conference.
The analysis, the way that we recommend preparing the DNA, actually ship us the frozen cell pellet with as little supernatant as possible and ship them on dry ice so they don't start degrading during the trip. Like Carolyn mentioned, the data analysis for all these techniques will be described in more details at the PCB annual meeting Bioinformatics workshop by Dr. Arno.
That will be next Friday, a week from today, in the afternoon in Dallas.
If you have any questions, I'll be glad to attempt to answer them.
Hi, this is Elias Zambitas from Baltimore. How you guys doing?
Hey.
I have a question.
Yeah.
Has left the conference.
Still here. So I have a question. You guys showed a protocol at the beginning of the show where you're basically enriching using a poly-A enrich strategy mRNAs, and that's standard protocol, right?
Yes, correct.
So I guess some folks have thought that poly-A negative RNAs may play a minor but maybe important role in pluripotent cells. Do you guys also have a protocol where you do RNA-seq of both the poly-A enriched fraction as well as the flow-through that might go through?
So we don't do that for in a routine basis. So until we have a good way of doing this, unfortunately, the balance here is in the availability of the library construction kit, the cost associated with it, and because we've put in some information in here. So currently, in order to maintain the low cost for the consortium members, we aren't planning on switching away from the poly. But I understand that scientifically, it would make sense to try to capture important messages that are not polyadenylated. So the answer is currently no.
One of, just philosophically speaking, one of the main goals for doing RNA-seq at all is to capture information qualitatively, not just quantitative, but qualitatively. That would be different than your typical really, really good gene array. So a lot of people might want to do RNA-seq to get RNAs that a gene array could not capture, not just quantitative but qualitatively. And then the other question I have for you is, if we wanted to do both microRNA-seq and messenger RNA-seq, then is that basically double the charge, or does it go?
We do them separately. It's I think 300 or 250 for the microRNA and 350 for the mRNA-seq.
Okay.
Depending on the reads.
Depending on the depth of read, yeah. Because the protocols are so different.
Right.
To get back to your first question, there is something that we can offer, which is the double-strand specific nuclease-treated mRNA-seq libraries, where we would actually not do any oligo-DT enrichment, but we would prepare the library from the total RNA, including the ribosomes, and then once after reverse transcription, we would denature the library and let it reanneal for 5 hours, which would then reanneal all the very repetitive parts of the transcriptome, which is mostly the ribosome RNAs, and treat them with double-strand specific nuclease after 5 hours, which would separate basically the two adapters from each other by cleaving any very abundant RNA in the sample. Because we have to re-amplify by PCR to differentiate the ones that have been cleaved from the ones that have not been cleaved, it becomes a not so quantitative method of doing mRNA-seq, but it is very effective at normalizing the amount of all the transcripts in the cell. So if you're interested in finding out in a quantitative way, we can accommodate you with the DSN protocol.
Okay. The data that we currently have in the consortium for the lines that Dr. Lutzko has coordinated is primarily mRNA-seq. Is that true?
Correct.
There's also a large batch of microRNA-seq and methylation data that is just coming off. Right. So there is and copy number variation. All of those are coming off.
Awesome. That's what I was wondering about. Okay, great.
Guys, this is Bruce. I really like Elias's idea about trying to go deep on non-poly-adenylated transcriptome. And I think the way to do it, we out a couple really good RNAs that are extremely representative of the group, a subgroup of RNAs. You know, the absolutely best E, the absolutely best H9, the absolutely best iPSC, and maybe, you know, a couple of classes where, you know, we think we have some, say, blood versus fibroblast differences or something like that. And then in those samples where we have all those RNA left, and then Mehdi described a very deep poly-A selections approach.
Has left the conference.
Maybe even get strand-specific. I mean, absolutely get strand-specific sequence on it. And so we're not doing that for, you know, every single in the database, but we're doing it to really get this other dimension of information from samples that we, you know, have vetted based on the conventional mRNA profiling as being really good.
Effort. Yeah, I like it.
Let's do that as a mini proposal to augment the database with that, some samples processed like that. It's a no-brainer. It's an absolutely great idea, Elias.
Yeah, so I mean, maybe after Dallas, when we start to look at the data and start to think about it, that would be a good time to repower out and think about that.
Sure. It would also allow us to quantify really how many non-adenylated RNAs have we missed in the lines, you know, and at least evaluation if it's really relevant or not, or if we should completely switch the protocol to something that is more like on a Rebo minus type. But these methods, while they do work for capturing the entire non-ribosomal RNA transcriptome, they add significant cost because of the number of different RNA species that are captured that are not adenylated, you know, are in the process of being spliced. We recover a lot of intronic RNA, so this requires a lot of additional sequencing to get read depth for all these species.
Absolutely, it's an investment for sure. I mean, it's probably end up being $1,000 a sample, but I don't think that that's a deterrent. I mean, it's really an extra layer of very important information. We'll sterile RNA transcripts coming up from enhancer and that sort of thing. So, and there's a couple of other projects, Mehdi, that, you know, we'd like to do the same thing. So when you're ready to do a run where you're going to use the specific non-poly-A based labeling approach, let's just do a whole. Let's look at putting together a whole batch of that, and some, you know, significant fraction will be from the PCBC samples.
Okay.
Let's now.
All right then. Well, thank you very much for attending, and enjoy Dallas.
Yeah.
Very much, Mehdi.
You're welcome.
Has left the conference.